All right. Hello again, everyone, and welcome to part 2 of the Chapter 22 lecture. Last time we went through the lymphatic system and introduced the main ideas with the body defenses. And remember that you can divide all of your various body defenses, which is your immunity, your various forms of immunity, into innate or non-specific defenses, also known as non-specific or innate immunity, and then adaptive or specific defenses, and again, immunity can be substituted for defenses. Remember that the general idea, if I was to go back to this picture, is that pathogens are disease-causing microbes such as viruses and bacteria, and oftentimes pathogens, the bad guys, will have antigens, little ID tags on their surfaces of different molecular shapes. And so a non-specific defense can fight off any kind of pathogen with any kind of antigen, and you're born with it, and you don't really have to worry about being exposed before. You just naturally have that defense. Whereas a specific or adaptive form of defense or immunity means you have to have some sort of exposure to a specific strain of pathogen with a specific molecular shape of antigen, and then you end up generating a very targeted response and a very powerful response that is going to involve antibodies that are targeted against that specific antigen as well. So that's, again, just kind of an overview of what we really mean by these terms, non-specific and specific. Again, you're born with non-specific, but they're weaker. They're not as powerful. They're not as targeted. On the other hand, with your adaptive or specific defenses, you're not born with them. You need to have some sort of exposure. You need your T and B cells to initiate the response. There can be a little bit of a lag time. It'll be highly specific and targeted and powerful. But if that pathogen mutates and its antigens change, and you don't recognize that specific antigen again, you have to go through the whole process of exposure and activation. So that would be the downside of something like specific immunity. All right, so what we're going to spend most of this particular lecture on are the innate or non-specific defenses, the ones that you're born with and that are very versatile. So an overview of these will include, first of all, just try to keep the bad guys out, have some barriers against them, such as intact skin as one example. And we'll be going through all of these in more detail shortly. We already know what phagocytes are. They're little white blood cells that do phagocytosis. They eat bad guys and other bad things. So those are very non-specific in their response. We've already learned a little bit about natural killer cells and their function, which is immune surveillance. So we'll take a closer look at how that works in just a few minutes. Remember, this is going to be targeted against bad cells in a very non-specific way. Then we'll get into these little chemicals called interferons, which have a nice antiviral property to them. We'll finally talk about the complement system. We've hinted at this before. It's kind of complex, but I will simplify it as much as I can, and we don't have to know all of it. Complement is basically a protein defense that is named because it complements or kind of helps alongside other things such as antibodies and phagocytes, so it helps them do their job. Inflammation, we've definitely talked about this a lot of different times. It's basically just your tissue's response to local damage, and you need a little bit of inflammation to kind of help the process along. I should say the healing process along, but too much inflammation can sometimes be problematic, which is why people sometimes will take anti-inflammatory medication. And then finally, fever. Whereas inflammation was more of a localized response, fever is more widespread throughout your whole body. You can kind of think about fever in a way as inflammation for your whole body. And you all know what fever is. It's just when you have a higher than normal temperature. It's a regulated elevation of your temperature, and we'll see how that helps in just a little while. So let's start with physical barriers. And of course, things that are associated with those barriers and things that are secreted from those barriers. The main barrier is just a nice, intact epithelium. So some epithelial tissues are thicker, such as, like a stratified squamous epithelium. Your skin is a keratinized stratified squamous epithelium. Your skin often will also have hair at the surface, so that's an additional barrier. Epithelial tissues are held together pretty strongly. You might recall from last fall in chapter 4, desmosomes are cell-to-cell junctions that really strongly attach these epithelial cells together. Some epithelial tissues have tight junctions, which prevent any leakage of anything between the cells, which is kind of nice. Underneath the epithelium, you can't really see it in this picture, but you have a fairly dense fibrous basement membrane. So if something was to breach the epithelial cells, it would still have to kind of get through the basement membrane before it could kind of get down deeper and cause an infection or get into your bloodstream. Many of your epithelial tissues will produce acid as part of their metabolism, such as, for example, we have an acidic pH on our skin, a very acidic pH in our stomach. We talked last quarter in the reproductive systems about the acidic environment of the vagina, and all of these can make the environment inhospitable to many pathogens that would want to infect you. And then some bodily secretions, including tears and saliva, have other antimicrobial properties, not necessarily low pH, but maybe other chemicals that are antimicrobial, and may or may not also contain antibodies to help fight off any potential pathogens as well. All right, mucous membranes will include, the lining of your respiratory tract, the lining of your digestive tract. So mucus is usually sticky, especially respiratory mucus, and that can trap pathogens that are in the air that you might have inhaled. And then there's you might remember this, a pseudostratified ciliated columnar epithelium that lines much of your respiratory tract that can sweep the mucus towards your throat so you can swallow it and destroy it and get rid of it. So mucus can trap bad guys, the cilia can sweep them away. And then other secretions are constantly just flushing over surfaces. Tears are constantly flushing your eyeballs. Saliva is constantly flushing your mouth as you're swallowing. Every time you urinate, the urine will flush out your urethra, the lower part of your urinary tract. So any pathogens that are trying to take hold in those places are constantly getting, a current of fluid trying to wash them away. All right, so this is first line of defense, right? Physical barriers, just trying to stop pathogens from getting inside your body in the first place, or making it very difficult at least. All right, next up for non-specific defenses, phagocytes. So some are smaller, so these are macrophages. This will include cells we've learned before, like neutrophils and eosinophils. Others are larger, so macro, means bigger. Monocytes are in the blood. Once they've left the blood, they can go out and be called macrophages. Ones that don't move, ones that stay stuck, are called fixed macrophages. Last quarter in chapter 14, you might remember the microglia. These are in your central nervous system. They just kind of always stay in your brain and spinal cord in case there are any pathogens that make it all the way in there. They can hopefully eat them up. Another type of fixed macrophage we're going to learn about next chapter when we get into the digestive system will be the stellate macrophages that are in the liver. So they always just stay in the liver. But other macrophages are mobile. So these are called free or wandering macrophages, and these are the ones that are capable of traveling just about anywhere. All right, some of these terms we've seen before. Emigration means that a white blood cell is leaving its homeland of the blood to go out into the tissues. It's also called diapedesis, kind of a process of walking through the wall of the blood vessel. So phagocytes, especially again these free macrophages, are capable of doing that. Remember the term chemotaxis means to respond to chemical stimuli in some way. If they are attracted and go towards the chemical stimuli, that's positive chemotaxis. There's also something called negative chemotaxis if they go the opposite direction, but mostly we're going to see examples of positive chemotaxis where a phagocyte is going to travel towards where the problem is because it'll be following the chemical trail that has been given off by the damaged tissue or the infection. So here we see a picture of a phagocyte that has kind of reached out little cytoplasmic extensions and made contact with lots of bacteria. So we would say that this phagocyte has gone through adhesion. It has attached itself to the pathogens that it's about to eat. And then of course what you have to do to finish this whole thing is to eat them. So phagocytosis, and then once it's inside of the cell, you destroy whatever you've just eaten with like digestive enzymes from a lysosome, oxidative enzymes from a peroxisome. Those are kind of the two most common ways to actually kill the pathogen once it's been consumed. And here we can see a free macrophage just getting ready to chomp down and surround some sort of little bacterium there. All right, so those are phagocytes. Those are non-specific defense cells. We've already talked about NK or natural killer cells being non-specific defense cells. Here's a little bit more detail about how they actually do their killing. So again, these are non-specific. So if these NK cells just bump into any abnormal, bad cell, here are a few examples here, it'll recognize it as being foreign or bad. Doesn't matter which strain. This is not a specific response. But when it recognizes it as being generally a bad cell, it'll kind of line up its secretory organelles and release these little chemicals called perforins. Here's a close-up view. Here's a secretory vesicle filled with perforins. And then there's your exocytosis. These perforins will actually latch onto the target cell's membrane and spontaneously form pores, little holes. Now when you form these little pores, the bad cell that has been targeted here can no longer maintain osmotic balance. There's no way to keep water from really rushing in. Generally, most cells are hyperosmotic. So if you put a bunch of little pores in a cell membrane with no ability to regulate anything going in and out, water will tend to rush in towards the solutes. So if water rushes in, it's going to swell up until it bursts. And remember, that's called lysis, when something ruptures or bursts. So that's how natural killer cells do their killing, is through basically making them unable to maintain osmotic balance. They fill with water and rupture. Pretty ingenious. All right, our next non-specific defense are interferons. And interferons are a type of chemical called a cytokine. Just in general, cytokines are chemical messenger proteins. Most of them act locally. Some of them can kind of get into the bloodstream and be more hormonal in their effect. But for our intents and purposes, we just need to understand that cytokines are little cell signaling chemicals, and your immune system uses them quite a bit. Interferons are one specific example of a type of cytokine. So interferons, as you can see, are released by some good guys, some lymphocytes that have become activated, and then some non-specific macrophages as well. And then even a cell that has become bad, one of your own cells that has become infected with the virus, can release these interferons. And they get their name because they interfere with viral reproduction. So looking at the picture down here, this cell is releasing little interferons. They will travel to nearby cells and cause those nearby cells to produce antiviral proteins, kind of protecting nearby and slowing the spread of the viral infection. It won't completely stop a viral infection, but at least it slows it down by protecting some of the most nearby or closest cells. As a bonus, these interferon cytokines will also act as positive chemotactic agents to attract other helpful non-specific cells such as natural killers and macrophages. So macrophages can attract other macrophages by releasing things like interferons. All right, so moving on to our next non-specific defense, the complement system. So there's a lot. You have, oops, messed up there. There we go. So you have a ton of different proteins normally just floating around in your plasma, and we definitely do not need to know them all. But you can see why they're called complement, because again, they kind of just help or enhance other things such as antibodies or other non-specific cells. There are different pathways to activate complement. If this is sounding kind of familiar, let me just quickly take you back to chapter 19 when we were learning about coagulation. Remember, there were a lot of different proteins, not as many as in complement, 11 versus 30, but still there were multiple pathways that were activated. We learned about the extrinsic pathway and the intrinsic pathway and the common pathway. Well, back then, I just had you know what began the pathway and how the pathway ended. That's the same approach we're going to take for complement. So let's take a look at these three pathways. We have the classical pathway, and I've got a picture of this on the next slide, so hopefully that will help illustrate, but let's go ahead and go through the words first. The classical pathway was the one that was originally discovered, hence classic or classical. So you can see it's also the most powerful and fastest of the pathways. And that's where antibodies are already attached to the bad guy. And then complement protein C1 recognizes that and kicks off the pathway. There's a bunch of in-between steps we don't need to know, but the end result is the formation of a complement protein called C3b, and it's attached to the bad guy. And again, I've got pictures of this on the next slide. But let's go ahead and take a look at what begins and ends the other two pathways. The lectin pathway, as you might guess, begins with a protein called lectin. Lectin can recognize certain carbs that are in, for example, types of bacteria that are kind of unique to bacteria. And so when lectin binds to those, you kick off a little bit of a pathway, and the end result is once again C3b. You'll see a theme here. The third and final pathway is the alternative pathway. This pathway was discovered well after the classical, so they're like, oh, here's the original, oh, here's an alternative, and then later they found this third lectin pathway as well. The alternative pathway is also known as the prepurdin pathway because the chemical prepurdin is one of the first chemicals that really kicks it off. Technically, it's an interaction between prepurdin, or factor P, along with factor B and factor D, that all work together. P, B, and D. That reminds me of PB and J, but instead of peanut butter and jelly, I just try to think of peanut butter and destruction. Because once you get this pathway going, one of the effects will be the destruction or lysis of the pathogen. So PB and D, peanut butter and destruction. But look at the end result, C3b. So all three pathways end with the formation of C3b, as we will see on the next slide. So here we go. And again, I apologize, this is kind of a big figure with tiny font, but if it helps, you can always just look at this figure in your book to get a different view of it. So let's just overview what we just talked about. Here's the classical pathway. Antibodies are already stuck to the bad guy. C1 recognizes that. You get this whole chain reaction that we don't need to know about, but the end result is C3b is attached to the bad guy. Lectin pathway. The protein lectin recognizes certain carbs that are in certain bacteria's membranes. Chain reaction. C3b attaches to the bad guy. And then the alternative pathway, also known as the prepurdin pathway. So there's prepurdin, factor P, combining with factor B and D, so PB and D. Chain reaction, C3b attached to the bad guy. And then when the C3b is attached to the bad guy, you can form these things called MACs, membrane attack complexes, kind of similar to what we talked about a few minutes ago with perforins. These complement proteins form a pore. Again, it's called a MAC because they're different proteins than perforins. They're just acting in a similar way. Once again, it can't maintain osmotic balance. Water rushes in, it swells up, and it lyses. It ruptures. In addition, complement can cause additional histamine release to increase inflammation, which remember, inflammation, a little bit goes a long way. You do need some, so this can be a good thing. That will also attract and activate phagocytes that might be nearby. So we're attracting some additional phagocytes to the area. So these are all things that C3b is doing. And then finally, you might remember from the last lecture, we defined this term opsonization. It basically just means kind of putting a handle on the bad guy. And so when you attach something to the surface of the bad guy, it just makes it easier for a phagocyte to come along and grab onto it and eat it. So enhanced phagocytosis through opsonization. Just, again, putting a handle, so to speak, on the surface of the bad guy to make it easier to grab onto and eat. All right, so that's the key core stuff that we need to know about complement. Definitely do not worry about every single step and every single numbered complement protein you see on all of these figures here. Okay, moving on. Speaking of inflammation, again, inflammation is a general, just kind of local response to any sort of damage to your tissues. So again, considered to be non-specific. It could be physical trauma like impact causing a bruise. It could be like more of a scrape. It could be harsh chemicals damaging your tissues. It could be something that is invading your tissues and causing an infection, such as a bacterium or a virus or a parasite or a fungus. It could be a heat injury like a burn. It could be a cold injury like frostbite. Whatever is actually injuring or damaging the tissue will cause this local response of inflammation. It's been a while, but way back in the fall, in chapter 5, towards the end of the skin chapter, we did learn a little bit about inflammation as the first phase of skin repair. So these are the things we saw back in ANP1. Cardinal signs, redness, heat, pain, swelling. You get some blood clotting to wall off the area, prevent the loss of blood, kind of, prevent pathogens from spreading. There's vasodilation, so you're bringing more blood flow to the area, which is going to bring oxygen and nutrients and white blood cells. The blood vessels become more permeable, allowing edema, allowing blood plasma to leak out more and flood the area with, again, oxygen and nutrients. And that's what's creating the swelling with the increased vessel permeability. So you've got all sorts of white blood cells coming to the area, and we're already starting the repair process. So these are the things we talked about with inflammation back in the fall, and you can see that more details were going to come when we got to chapter 22, and here we are. So nothing really brand new. You get the damage, you release histamine. Mast cells, remember, are basically just like basophils that are out in the tissues. They're filled with little granules that contain histamine and heparin. So that histamine is the most powerful chemical for being a pro-inflammatory chemical. So it's that histamine that's going to cause your vasodilation, your increased vessel permeability. You're going to stimulate the clotting process so that, once again, you get a little temporary repair and try to wall off the area. All this blood that's coming in and leaking out is causing your redness and your heat and your swelling. And the damage has irritated nerve endings, and chemicals have been released that stimulate pain receptors. So you're feeling the pain. White blood cells are being attracted to the area. And once they get there, they are going to not only attract additional non-specific defenses, such as neutrophils, macrophages, and start the tissue repair process with fibroblasts, but you will also hopefully get some specific cells like some T cells and some B cells to come along to start a specific or adaptive immune response. But the bottom line is, again, you need all of this, at least in moderation, to really get that permanent tissue repair going. So if we were to summarize, There's our symptoms. We get a temporary repair. We try to wall off the damaged area. Both non-specific and specific cells and defenses will be activated. And then the hope is that you transition from this temporary repair to more permanent repair down the line. And this is another way to kind of summarize it, all those different things we just kind of talked about over in this flowchart. Okay, so again, inflammation is more of a local response, whereas fever, which is the last of our non-specific defenses we're going to talk about, is more of a widespread, body-wide response. A regulated elevation, which means your brain is doing it on purpose. Your hypothalamus, which is the brain's thermostat, resets to a higher temperature to make you feel cold. That's why you often have the shivers and the chills when you have a fever. I mean, you're burning up. Somebody feeling your skin would be like, whoa, you're hot. But you feel cold. That's your hypothalamus playing a trick on you. So that causes your temperature to rise because your body thinks that, oh, you got to get up to this new normal that your hypothalamus has set it at. And the chemicals that are going to tell the hypothalamus to start a fever process are called pyrogens. I love this term. Pyro, fire, gen, create or start. Fire starters. Basically, that's what pyrogens are. They could be endogenous pyrogens that are made by you in response to fighting off whatever's infecting you, or the pyrogens might actually be chemicals that are released by the pathogens themselves, as opposed to ones that you made. Regardless, pyrogens cause you to have this on-purpose raised temperature that, as long as it doesn't get too out of control, because if you get too hot, your whole body can start shutting down. But a mild fever will boost your metabolic rate, which means that your white blood cells and other defenses are going to be able to act more quickly and be more efficient in fighting off the infection. And many pathogens are adapted to reproduce most efficiently at your normal body temperature of maybe, close to 98.6 F. So if you crank it up to maybe like 101, 102, they do not like that very much, and that will actually inhibit certain viruses and bacteria. So that seems to be a nice little benefit of a mild fever as well. All right, that is a wrap on the non-specific defenses. And the last part of this particular lecture that I wanted to do is just at least introduce the adaptive or specific defenses. We won't get into the details of them in this lecture, but let's just make sure that we're feeling good about this terminology again. So again, the word specific and the word adaptive are pretty much interchangeable in this context. As we've talked about before, you need T cells and B cells to have adaptive or specific immunity. So again, how many times again can I say the word specific? But like we talked about before, here's the picture again. Specific or adaptive immunity is targeted to antigens of a specific molecular shape. So it's just like with surgeon-like precision, can you go in and try to kill and neutralize pathogens. So it's very targeted, very specific. I mean, it's right in the name, right? Specific. It's very versatile. So even though you have never been exposed to most of the possible antigens that are out there in the world, you have the capability of recognizing them. You have millions of lymphocytes, different types of lymphocytes in your body right now that are just floating around inactive, and they won't become active unless they encounter the specific antigen that they are capable of recognizing. So that's pretty incredible that, you may not ever come across a specific antigen of a specific disease, but if you ever do, you do have the right type of lymphocyte to be able to recognize it and start responding against it. So again, very versatile. Memory is an amazing aspect of adaptive immunity. When you have an adaptive immune response, the first thing that's going to happen is you're going to have lots and lots of cell division, and those active cells that respond immediately, they're trying to keep you alive. They're trying to, fight off the bad guy. But then some of the cells kind of hang back and don't actively fight or respond. Those are the memory cells. They stay inactive and they can hang around for months, years, sometimes even decades. And if you ever encounter that exact same specific antigen again later on, those memory cells can very quickly activate, and you can often wipe out that second uh, infection before you even realize you got exposed. I mean, it's that efficient and effective. Again, the downside of memory is if there's a mutation, if the antigen shape changes between the first time you were exposed to it and then future times, you may not recognize it the future times, and you might have to undergo the entire activation process all over again, which takes a lot longer. All right, and the last property here is tolerance. If everything's working correctly, if your immune system is on point, it should not react against your own antigens, right? Your self antigens. However, sometimes tolerance breaks down. I'm sure you've heard of autoimmune disorders, things like lupus and type 1 diabetes and multiple sclerosis and what was the other one I'm thinking of? Rheumatoid arthritis, just to name a few. I mean, there's dozens of them. Those are all examples of where your immune system falters to the point of it's attacking your own cells, your own tissues. Again, normally that shouldn't happen, but if you have an autoimmune disorder, it is happening. So the property of tolerance is important, but not always perfect. Okay, so some more terminology here in terms of forms of immunity. We've already talked about the innate or non-specific defenses over here that you're born with, genetically determined, you don't need exposure. But on the downside, there's no targeted specific antibody production or anything like that. But now we're kind of focusing more on the adaptive or specific stuff, where there is going to be antibodies involved. You're not born with it. There's going to need to be some sort of intervention that happens. And that intervention can happen actively or passively. This is from the perspective of you as the patient. So active means, if I'm talking about me, active immunity in me means I am exposed to the antigen and I'm going to make my own antibodies. So the natural way for this to happen is just to get sick. So if I get exposed to the bad guy's antigens and then I develop my own antibodies against them, I have just done naturally acquired active immunity. I've gotten sick. Now a shortcut to all that is a vaccine. That's artificially acquired active immunity. Instead of me having to get sick to get exposed to the antigens, I can have the antigens administered to me in a vaccine. This could be dead or weakened pathogens. This could be isolated antigens that have been engineered to not make me sick, but it still counts as an exposure. I'm still going to, in response to that, make my own antibodies. So that's why it's active. So that's artificially acquired active immunity. I'm making my own antibodies with both of these over here on the left. Now over on the right, passive means, from my perspective, I'm not making my own antibodies. They've been donated to me from another source. So naturally acquired passive, the best example is going to be from mother to baby. So even before the baby's born, some antibodies can cross the placenta. From the baby's perspective, the baby didn't make those antibodies. They came from a donor. They came from mom. After the baby is born, antibodies that are present in breast milk are being donated from the mom to the baby. So from the baby's perspective, it's passive because the baby is not making his or her own antibodies. So that's naturally acquired passive. And then artificially acquired passive would be just basically an antibody shot. So what if you didn't, for example, ever get a vaccine against rabies, but you get bit by a rabid animal? It's kind of too late right now to get a vaccine and build your own immunity. You just need a quick injection of antibodies that are targeted against the rabies virus to try to cure yourself, short term, right away. So in this context, if I got bit by the rabid animal and I got a rabies antibody shot, I'm not making those antibodies. They have been donated to me, through the needle, administered to me. These passive immunities are not long lasting because as soon as those antibodies kind of wear out, remember, they're just proteins. So molecularly, they're eventually going to wear out. So this is more of a short-term fix or a short-term type of immunity, which is passive. But active immunity, because you're making your own antibodies, can last much, much longer. Sometimes years, sometimes even longer than years, decades. Okay, so hopefully those terms make sense. So the last thing I want to do with this lecture is just make sure that we're understanding this overview of the adaptive immune response and that it has these two major arms, cell-mediated immunity and then antibody-mediated immunity. So we've already talked about this a little bit, but as the name implies, cell-mediated means we're going to go after bad cells. Here are all the examples we've talked about before of cells that are bad, that need to be taken out, killed, controlled in some way. Now the cytotoxic T cells, remember these are sometimes called killer T cells, these are going to specifically target those bad cells. But helper T cells, as I mentioned before, they're lovers, not fighters. So they will release cytokines, cell signaling chemicals, that will cause some non-specific cells to get involved, which will help other types of T cells mature, such as the cytotoxic Ts. And then this is a key link here. You need active helper Ts to activate the Bs, which is the other arm of immunity. Okay? Helper T cells are the link between cell-mediated immunity and antibody-mediated immunity. Now remember, humor, like think aqueous humor, vitreous humor, it means fluid. So we tend to have antibodies in our body fluids such as blood plasma and saliva and sometimes mucus and breast milk and tears, just to name a few. So antigens that are either floating freely in those fluids or are attached to pathogens that are floating in that fluid can be attacked by antibodies. And so, like we mentioned before, B cells, when activated, will grow and differentiate into larger cells called plasma cells, and it's the plasma cells that then release the antibodies. So here's kind of a cartoon view of that. And here's your two arms. Here's your cell-mediated arm, and here's your antibody-mediated arm. So you need some sort of exposure. Here are some pathogens. Look at all these little surface antigens, all these little dots on the surface of these bad guys. So it's probably going to start up here with, something like a phagocyte coming in contact with the bad guy, consuming, killing it, doing antigen presentation. T cells will be activated. This will include both cytotoxic T cells and helper T cells. The cytotoxic T cells will actually do the attacking and killing of the bad cells to destroy them. Whereas the helper T cells, they're more just communicators. They're coordinating between the two and activating B cells to make sure that you get the antibody-mediated arm of immunity going as well. And again, B cells will become plasma cells will release their antibodies, and then the antibodies will actually carry out, again, additional destruction of bad guys. All right, so that's a good overview of adaptive immunity. That's a great place to stop for this particular lecture. As always, hopefully as questions come up, you write them down and then you can get them answered. Just email me, drop into a Zoom session, and we can talk about it. All right, that's a wrap for part 2 of the Chapter 22 lecture. Next time, we will continue on with the details, and they're pretty fascinating, of how adaptive immunity works. And that will wrap up Chapter 22. All right, thanks for listening to this one. Have a great day.